All right, so for some reason the video cut off last night, probably because I don't know what I'm doing with the camera, so I'll finish it this morning. So I was talking about at the end the alpha 2 receptors, which are part of the sympathetic nervous system. They also act on a lot of glands. I told you before that the insulin, when they're activated, the postganglionic nerve releases norepinephrine, and that binds to the alpha 2 receptors. A lot of them are on the pancreas, which is a really good example. It's inhibitory, so it's going to decrease the amount of insulin produced. Thus, we know that insulin, when there's a lot of it circulating in the blood, that glucose is taken up by the cells and can be stored for energy for later use. So that process, so thereby it gets taken up into the cell and used and made into glycogen, is called glycogenesis. And since we want to do the exact opposite when we're under our fight or flight response, we want more glucose into the bloodstream so that our skeletal muscle can use it to run away or fight something. We are going to have a decrease in glycogenesis. And then we're going to have an increase in glycogenolysis. So insulin is responsible for both of those actions, just the decreasing of it. Alright, so then we've got our beta 1 receptors. Now they're excitatory. They're found in the kidney, the stomach, and the heart, like I put there. In the heart, they're going to increase contractility, so they're going to make the heart beat harder. They're going to induce the heart to push out more blood with each pump, which is going to increase blood pressure too, to some degree. We'll go over Starling's law of the heart later on, I think. So it's going to increase contractility, make the heart beat faster, harder, all that kind of good stuff that you'd expect from a fight or flight response, right? Okay. It's also going to work on the kidneys to increase renin secretion. So renin is part of the angiotensin-aldosterone system, or RAAS, renin-angiotensin-aldosterone system. And it is going to increase renin secretion. So there's these cells in the nephron of the kidney called the juxtaglomerular cells, and they are influenced by the beta 1 receptors from the postganglionic neuron, which is releasing norepinephrine, to secrete renin. Okay, so renin functions through a really or not that complex a mechanism, but it's going to tell the body to create this thing called angiotensin 1, which is going to be cleaved into angiotensin 2, and thus influence blood pressure to rise. So there's a drug that acts on this system called ACE inhibitors. So ACE is this angiotensin converting enzyme, and when you block that, you get a decrease in blood pressure. So that's kind of a good way to remember what their renin-angiotensin-aldosterone system does. Interestingly enough, it's also going to work on the stomach, and it's going to increase ghrelin. So ghrelin and leptin are two antagonistic hormones. They do the opposite things of each other. Increases in ghrelin are going to actually make you feel hungry, which is kind of odd, which you wouldn't think, because the parasympathetic is more of a rested digest kind of thing, but it is only active after you eat. So you got to get food first. Okay? Beta 2 receptors are inhibitory. I also call them another catch-all receptor, just kind of like the M3, and they are also on smooth muscles. Okay, so they're just like on the M3 of the parasympathetic. It's going to induce smooth muscle to relax, so norepinephrine binds to these receptors. They're all over in the smooth muscle cells of our body. And where is smooth muscle in our body? Pretty much everywhere. So it's in primarily our gut and our lungs, also in our vasculature. But the main one I wanted to point out was the lungs. So we said here that the M3, once they're initiated or excited, that they induce smooth muscle contraction. So I said that in the lumen of the bronchioles, they would be decreased because smooth muscle, which wraps around the kind of piping of the lungs, it contracts and clamps down on the airway, so it makes it much smaller. Well, this is going to increase the lumen diameter. So it's going to relax. Since it's inhibitory, what's it going to do? It's going to make something, stop something from doing something. So it's going to stop it from contracting. So the smooth muscle relaxes when they're stimulated by the beta-2 receptors. A good example he gave for this in class was asthmatics. When you need to, when they have an attack, an asthmatic attack, their lumens are being, in their bronchioles, are being clamped down on, being very small, so it's very hard for them to breathe. And the way to get around that, or the way to cure that, fix it, is you give them, well, the primary one is albuterol, or duo-neb treatments, and those are responsible for binding to the beta-2 adrenergic receptors. And when that happens, it induces the smooth muscle relaxation and it opens up your airway again so they can breathe. So that's a really good example, and I really like that example for beta-2 receptors, is that they go ahead and relax the airway. So albuterol is a good way to remember the beta-2 receptors. Alright, so our GI receptors, or our GI beta-2 receptors, are also going to induce smooth muscle relaxation, so they're inhibitory, right? They are going to relax smooth muscle in the alimentary canal, and basically they're going to decrease gastric motility. So that's a fancy way of saying everything's going to sit in your gut, just kind of like after kind of a heavy meal, or if you're going on the plane when you're going on vacation and you I don't know if this happens to some people, but if you go far away, you get really excited, there's a ton of build-up for your trip, and you're like, then you get there and you're constipated for like a couple days. Well, that's because the beta-2 adrenergic receptors are inducing smooth muscle relaxation, so all your food that's in your like small intestine and large intestine is just sitting there because the muscles are not contracting and pushing it along. So that's a good way to remember that in the alimentary canal. Alright, so the last two here that are kind of a part of the adrenergic receptors, but they're not technically, even though dopamine is cholinergic, or not cholinergic, excuse me. They are an adrenergic. They are part of this family of molecules. So norepinephrine, dopamine, and epinephrine are part of our catecholamines class of molecules. So they are grouped in with the sympathetic nervous system, but they are not technically a part of it, if that makes sense. I'll explain a little bit more. He didn't really go over these that much, but the dopamine receptors, I think the main thing we got to know is the dopamine 1 receptor is excitatory, the dopamine 2 receptor is inhibitory, okay? And they're mostly in the CNS, okay? And the other one place they are is in the kidney, which is going to become a little important later on for like certain kind of emergency medicines that we can give. But in the CNS, they're going to be really responsible for reward. So reward functions, they're going to make you feel happy, and it's basically the source of addiction is the dopamine release that you get from after doing like drugs like cocaine. It's a good example of this because that's why he was talking about, do you like cocaine? Alright, so that's good and well, that's all the receptor types, okay? Now how does each of them work? How does each of them, when they stimulate the effector organ, and when these neurotransmitters bind to these receptors, what do the receptors then do on the cell of the organ to make the change happen? Well, we have 3 different ways that it makes it happen. So I've outlined them here. Mechanism 1, 2, and 3. Okay? So mechanism 1, these are all going to be second messenger systems. Okay? As opposed to in the somatic or in the preganglionic neuron, where they were just binding to acetylcholine and inducing the change of just the ion flowing through, of sodium flowing through. So in the preganglionic and in the somatic nervous system, we had acetylcholine binding to those receptors, and they were basically sodium ion channels, so they just allowed an influx of sodium. Well, these, they're going to be mediated by a G protein, which is a chemical second messenger system, which is just a fancy way of saying this isn't an ion channel that it's acting on right away. It's going to send something, the receptor is going to send a little molecule to do something else, so that way we can get a different effect, rather than depolarizing the cell, which you don't always want to depolarize the cell. You want to make some different changes. It's another way to do that. So in the effector organ, we're going to have chemical second messenger systems, and they're going to be G protein mediated. Okay. Just a little bit of review. G protein or second messenger systems are called metabolotropic, because metabolotropic means a metabolite of a reaction is driving the process. These are ionotropic because the sodium, it's just inducing the sodium channel to open up. Alright, so our first mechanism is mediated by this GQ G protein. GQ is going to be cleaved off our receptors. So in this case, we've got our alpha 1, M1, and M3 receptors that use this mechanism. And it's going to go and activate phospholipase C. Phospholipase C is going to do a bunch of different fancy things. There's these like IP3 and DAG proteins that it's going to act on. We don't need to know that per se, but what we do need to know is the end result of what phospholipase does. Phospholipase C does. So the end result is that it's going to increase calcium concentration in the cell, and it's also going to increase the activity of protein kinase C. Okay? The results kind of make sense, and what receptors use this mechanism makes sense if we go back through and kind of think about it. So with our mechanism 1, we're going to get an increase. I focus on the calcium. Well, what does calcium do in a cell that's really beneficial? Well, in smooth muscle, or pretty much all muscle, it's going to allow the fibers to contract, right? We need that fiber, or we need the calcium to basically let our muscle fibers slide past each other and contract. So what ones deal with smooth muscle? Well, we know our alpha 1s do, or we know our M1s do, and we know our M3s do. They all deal with smooth muscle contraction. Okay? So our alpha 1, like I said, in the arterioles, there's a lot of these receptors, and they induce the smooth muscle contraction to increase your blood pressure because it's the flight or flight response. They are also around the outer sphincter of your bladder and allow you to hold your pee. Okay? You don't want to be peeing while you're running away from something unless you're really scared. Alright, they're also in the M1 and M3. So the muscarinic M1 is excitatory, and it's going to increase your glands to secrete something. The good way to remember this is protein kinase C. Protein kinase C is going to make something do something. Okay? So increasing protein kinase C makes the glands secrete. Okay? And then the M3, which is a better example of this, why this mechanism is used here, is also in smooth muscle. It's going to increase your calcium levels so that you can go ahead and clamp down or contract your smooth muscle. So like in your asthmatic, it's going to be stimulated so that it can decrease the lumen diameter. Our mechanism 2 is used by beta 1 and beta 2 receptors. It's mediated by a G protein called GS. GS is going to go within the cell and through a series of different mechanisms that we don't need to know, it's going to turn ATP into cyclic AMP. Cyclic AMP is a chemical messenger. It's like a hormone of the cell that's going to increase. And when that happens, we get an increase in the protein kinase A. So why does the beta 1 and beta 2 receptors use this? Well, they're going to increase the contractility, they're going to increase blood pressure through the kidney, renin, angiotensin aldosterone system, and they're going to increase ghrelin. It's also going to work on the beta 2 receptors because it's going to increase the protein kinase A, allows smooth muscle to relax. Okay? And then we've got our oddball, our last one. We've got our M2 and alpha 2 receptors are kind of activated this way, or bring about change in their effector cells this way. They're going to decrease cyclic AMP because of this GI protein that's created when the receptor is stimulated. And the results of that is the protein kinase A is going to be downregulated, so there's not going to be as much of it in the cell doing stuff. So what receptor types work on this mechanism? The M2 and the alpha 2. So they're both inhibitory actions within their effector organs. We get a decrease in protein kinase A, so we get a decrease in insulin production. Decrease, decrease. Makes sense, right? In M2, we have a decrease in heart rate and a decrease in contractility because it's an inhibitor. That is caused by protein kinase A. So yeah, that's kind of the end of the receptors. Sorry that this didn't get recorded before. Couple things. Let's think of some stuff with, like, I'm going to go back through and make another video for the eye because that's a really good example of the dual innervation of an organ. So these two tracks, there's going to be a receptor from each track on an effector organ. There's going to be a parasympathetic receptor and a sympathetic receptor. And that's because it's like a shower. We need to get the desired temperature in our shower with our two knobs. We need to play with them until they get to the right temp. If we can't do that, or if we don't do that, it's not going to be the right temp. We're going to scald ourselves, or it's going to be a freezing cold shower. So it's better, it's much easier to get the right temp by adjusting two knobs than it is just doing one. Okay. So with parasympathetic, if we withdrew the parasympathetic response, I'm going to give the example of the M2 versus the beta 1 receptors because they're both kind of in the heart, and it's a good example. It's pretty easy to understand. If they're both in the heart, they're going to M2 is going to slow down or inhibit the heart muscles from contracting as much and as easily. And I think I kind of got into the mechanism for that in the earlier video. And the beta 1 receptors are going to do the exact opposite. They're going to speed up the heart and increase contractility. So the reason we have these two ones on there is because we can activate the beta 1 receptors to increase our heart rate, and at the same time we can inhibit our M2 receptors so that it doesn't get as much innervation and allows this to really work well. It'll increase the heart rate a lot better. Okay? And then the opposite, we could get the M2 receptors really stimulated, so it could really slow the heart down, way, way down. Okay? And at the same time, we could be stopping or blocking the effects of the beta 1 receptors. So that would decrease, it would work in tandem with the M2 receptors to decrease our heart rate and contractility. Okay? A good way of thinking about this is with beta blockers. So beta blockers are a really, really common drug in America. Beta blockers block the beta 1 receptor. And everybody, or most of us probably have kind of come across them in like our clinical experiences before this. The most common one that's prescribed is propranolol. All the beta blockers end in olol, that's the way I was remembering.
And per panel